cellular respiration can be broken down into a couple key phases. Glycolysis, and then it can go either two routes. The aerobic pathway through the Krebs cycle and the electron transport chain, or the anaerobic pathway using fermentation. We're going to be taking a look at glycolysis to begin with, and if we were to break down this word glycolysis, we have this first part of our word glyco. Glyco can be derived from our Greek word known as glycus. Glycus, what glycus means is sugar or sweet. It means sweet. And typically, sweet in biology, at least as far as I'm aware, is going to refer to a sort of sugar or polysaccharide. And in this case, it's going to be referring to glucose. In terms of the second part of our word, lysis, what lysis means is the breakdown. So we have the breakdown or / splitting of something. To break up or split something. So if we put these two terms together, we get glucose being split. And that's effectively all glycolysis is. It's the splitting / breakdown of glucose into usable forms of energy, or pyruvate. This process can occur without oxygen, making it anaerobic, or in other words, anoxic. From this reaction, we can proceed further into the anaerobic pathways, as mentioned earlier, which are either lactate fermentation, which produces lactic acid in humans, or in other organisms like yeast or bacteria, they will produce alcohol or ethanol, which we'll discuss later. But if we have oxygen, then we can proceed into our Krebs cycle, sometimes called the citric acid cycle. It's called this because it deals with citric acid, the same juice that's, again, used in lemons or oranges or sour food. From there, we move on to the electron transport chain. So glycolysis, we're going to focus on mainly glycolysis and the aerobic pathways. So we go in from glycolysis to our Krebs cycle, and then into the electron transport chain, and that is the aerobic pathway. That's when we have oxygen. And what happens in the electron transport chain, that's where the bulk of ATP is produced, using raw materials from all the other processes mentioned earlier. So now that we've actually taken a look at what glycolysis actually means and how the process actually begins to look like, remember that this whole process of glycolysis actually takes place within our cytoplasm of our cell. So it's going to take place within our cytoplasm, and it is going to be an anaerobic process. So we're not going to have any involvement of oxygen within our glycolysis. So it is an anaerobic process. So this can happen independent of oxygen, whether we have oxygen or not. So just remember that. And from here, remember, we start off with a glucose molecule. As we mentioned, glycolysis is effectively the splitting of a glucose molecule. So we're going to start off with our glucose. And glucose, we know its formula. It's going to be C6H12O6. And it follows that common 1:2:1 ratio that carbohydrates tend to follow, as you'll probably notice. But what we're going to be more so focused on is the carbon backbone of our glucose molecule. And what that means is we're just looking at the amount of carbons. So we have 6 carbons present, so each dot is going to represent the amount of carbons present within our molecule. And then from here, we can actually start our first process of glycolysis, our first stage. And our first stage is essentially, our goal is going to be to oxidize glucose. So in order to oxidize glucose, we're going to have to make an investment. And the first stage is going to be called, or I like to call it, the investment phase. So what we're going to do is we're going to invest a 2 ATP. So the cell is going to invest 2 ATP in order to break it down and create 2 ADP plus 2 phosphate groups. And what's going to happen with these 2 phosphate groups is they're actually going to attach. Each of these are going to attach on the end of our glucose molecule, and effectively it's going to attach to our glucose and make it high energy and unstable. It's going to be ready to break apart by this point, and we're actually going to get a splitting down the middle to create 3 3 carbon molecules with the phosphate group on the end. And that resulting molecule is going to be PGAL, something called PGAL. And if you remember from our Calvin cycle, you probably recognize this molecule with the 3 carbons and the phosphate group on the end, PGAL. Or in other words, it can be called, it takes many names, it can be called G3P. So now these modified glucose molecules are called either PGAL or G3P. And here's the key thing, from this point on, everything is going to happen twice, once for each PGAL. So past this first stage of investing the 2-ATP, you're going to have this process of glycolysis be replicated on both sides. So you'll see what that actually means. And you probably maybe are wondering, like, why? Why, if the whole point of cellular respiration is to create ATP, and why do we immediately use a 2-ATP to begin with? So the answer to that is because in the end, we need this energy to, again, oxidize glucose and create these 2 high-energy compounds called PGALs. We actually get more ATP in doing this than we do investing, so it's actually a net positive of which we're gaining ATP in the end. So that's why we actually do this. We're not actually losing any ATP, but you'll see what that actually means as we start to progress. Now, what our PGL is going to go through is a few steps where it gets oxidized, and it's going to lose electrons, and those electrons are going to be passed to a molecule called NAD+. So our PGL is going to lose electrons and get oxidized, and if you have to, like, there's no shame in always writing, Leo the line says GER, so Leo the line says GER. Remember that loss of electrons is oxidation. We're going to lose electrons from PGAL, and these electrons are going to be passed on to NAD+. So NAD+, and NAD+ is going to get reduced into NADH. So we're going to reduce NADH, and gain of electrons is reduction, remember that. So we're gaining the electrons lost from our PGAL to reduce NAD+ into NADH. That NADH is going to be an energy carrier, and we'll use it later in the electron transport chain. So as the PGAL continues through the pathway, it also produces 2 ATP individually. So we're going to produce from 2 ADP plus a phosphate, plus 2 phosphates actually, we're going to produce 2 ATP. And remember what I said about this PGAL, right? For each PGAL, everything is going to happen twice. So we're going to have this left side with the same process. So we're going to have the PGAL being oxidized and losing electrons, which are passed down onto NAD+, then are used to reduce NAD+ to NADH. Useful for the electron transport chain and creating ATP in the end. But also, this helps us reduce this ATP and create ATP in the end. So we're going to be resulting with 2 ATP on both sides. And you're probably wondering, what is the process, or what is the product of this process? So the product is going to be pyruvate. It's going to be called a molecule called pyruvate. So we're going to have 2 pyruvates for 1 glucose molecule produced. And what pyruvate is, it's a 3-carbon molecule, so 3-carbon molecule. And finally, each PGAL is going to be converted into the pyruvate, the 3-carbon molecule that marks the end of glycolysis. So, from 1 glucose molecule to begin with, we ended up, so we put in 1 glucose molecule, and what we got out of it was 2 pyruvates, 2 NADHs, which are used to create ATP in the electron transport chain, and a net 2 ATP, so a net of 2 ATP. And you're probably wondering, why is it a net of 2 ATP? Because remember that we've actually used 2 at the beginning in order to create 4 in the end. So we actually have a net profit of 2 ATP. We've created a total of 4, but we had to spend 2 in order to create those 2. So we have a net of 2 ATP. Another thing we also put in was 2 NAD pluses. So that was going to be our input. We have these 2 NAD pluses over here, which were inputted. And we had an initial input of 2 ATP. So in doing so, we created 2 pyruvates, 2 NADH, and a net of 2 ATP. This diagram really just lays out the input. The splitting of glucose into 2,3 carbon chains are known as PGAL, which are a letter used to output our pyruvate, and it demonstrates the energy exchange along the way. So we have the ATP being produced and our NAD plus being reduced into NADH for our electron transfer chain, and that's all glycolysis is. We're breaking down this high energy glucose molecule that has a lot of high energy potential, and we're turning it into pyruvate, which is later used in our Krebs cycle. So that's all glycolysis is. It's fast, efficient, doesn't need oxygen, and just a little energy up front to get it started. And boom, you've got the power to fuel the rest of the cell's energy cycle. So as we remember that, if I mentioned earlier that pyruvates are post glycolysis, we have this resulting molecule of 2 pyruvates, right? So there's 2 ways our pyruvate can go. It can go down the path where we have no oxygen, so anaerobic conditions, or we can go the path where we do have oxygen present. And that's where we have our Krebs cycle actually taking place. In this video, we're going to be strictly looking at the Krebs cycle and the electron transport chain, which is going to be the oxygenated pathway. So we're not going to be focusing on we're going to be more so focusing on our aerobic respiration rather than our anaerobic. But if you are curious, it's really short and simple. All we're doing during no oxygen is basically a process of fermentation where we recycle NAD plus for further glycolysis. But basically, we're going to be taking a look at the oxygenated pathway with the Krebs cycle. So in order to do that, we need to zoom in to a mitochondria's cell and actually analyze where it actually begins and takes place. So let's do that. If we are to zoom into a diagram of the mitochondrion, the Krebs cycle takes place within the inner space of this mitochondrion, so effectively the cytoplasm of this mitochondria. This is called the mitochondrial matrix. So this part right here, I know you can't see it, but is the matrix. Yeah, but basically, the Krebs cycle actually takes place within the mitochondrial matrix. These convoluted little flaps that maintain and increase the surface area are called the cristae, and is where the next process, the electron transfer chain, actually takes place. So from our process of glycolysis, we've actually produced 2 pyruvate molecules. We know this. These pyruvate molecules are going to be 3 carbons. So the carbon backbone is going to be 3 carbons long. Effectively, what we're doing here is, before our Krebs cycle, we need to actually prep and prepare these pyruvates and convert them into something called Acetyl CoA. And that's what initiates the process of the Krebs cycle. So we need to convert these pyruvates into Acetyl CoA. In order to do this, the first thing we need to have is this enzyme that actually helps us create this reaction and make it possible, called CoA. So coenzyme A basically helps us break down pyruvate, a 3-carbon molecule, into a 2-carbon molecule, Acetyl CoA. And you're probably wondering where does this 3-carbon electron, like where does the 1 carbon go from 2 to 3? So we're going from 3 to 2 carbons. Where is that released? It's actually released in the form of carbon dioxide. So carbon dioxide is going to be a 1-carbon molecule, and it makes sense, right? Because we have 3 initially, and we're releasing this 1-carbon molecule to turn it into a 2-carbon molecule known as Acetyl CoA. So acetyl coenzyme A, if you want to call it. Another thing we're actually doing in this process of oxidizing pyruvate into Acetyl CoA, we're actually reducing this molecule, again, very common, we're reducing NAD plus into NADH. And this is the same idea here. Remember that when we're oxidizing, we're losing electrons. When we're reducing, we're gaining electrons. So we're losing electrons from our pyruvate, and that's actually helping us with this reduction, because remember, to reduce, we're reducing NAD plus to NADH. We're actually reducing, and we need electrons. So we're going to gain electrons and gain a hydrogen, and these lost electrons from pyruvate actually help us do this. And this whole entire process is called pyruvate oxidation. And this actually takes place before the Krebs cycle. A lot of biology textbooks tend to skip out on this and actually include this and credit this to the Krebs cycle when it's not even a part of the actual Krebs cycle. It's actually a preparation phase. We're creating acetyl CoA so that we can have this molecule called acetyl CoA actually enter our Krebs cycle and be ready to start. So this whole process here is going to be called pyruvate oxidation. So now that we've actually created this 2-carbon molecule known as acetyl CoA, we actually need to consider is how the pyruvate actually goes from glycolysis to pyruvate oxidation to the Krebs cycle. So remember, from glycolysis, our resulting molecule was a 2 pyruvate molecules, and in order for them to actually be sent to our pyruvate oxidation, remember that oxygen needs to be present. This is the anaerobic part, the anaerobic pathway of cellular respiration. So we need oxygen to be present so it can be actually transported. And if there is oxygen present, so with O2, what basically happens is our pyruvate is going to be now sent to our mitochondrial matrix. So we're sending it back to the matrix. So this is where pyruvate oxidation is actually carried out, and also the Krebs cycle is actually carried out. So remember that I mentioned earlier in that the diagram of the mitochondrion, the Krebs cycle is going to take place within the matrix, and so does the pyruvate oxidation. So don't get it twisted. Remember that the only thing that's taking place within the cytoplasm is actually going to be glycolysis. So now to get into this first step of the Krebs cycle, effectively what's happening here is acetyl CoA is going to come in. So we've generated this 2-carbon molecule, as we know, acetyl CoA. And what we're doing in our first step is we're going to be regenerating our coenzyme A. Remember, we use this coenzyme to break down pyruvate into acetyl CoA. So I'll highlight this here. This enzyme called CoA. So all we're doing in the first step is of the Krebs cycle is we're regenerating this CoA. So what's going to go out is going to be CoA in our first step. From here, we have step 2. We have a 4-carbon molecule that's where the Krebs cycle actually takes place. So our 4-carbon molecule would be like the RUBP equivalent of the Krebs cycle. We have this molecule that we need to be added to our input, which is acetyl CoA, and in the case of the Calvin cycle, we would add the RUBP to the carbon dioxide to form a 5-carbon compound. But in this case, we're adding a 4-carbon compound to a 2-carbon, and the resulting 4 plus 2 gives us 6. So we're going to have the 6-carbon compound here, and effectively what it's going to be called is citric acid. So citric acid, what citric acid is it's the same juice that's in your oranges or in citric. So I don't know why we wrote citric juice there. Citric acid. And this is the same juice that's in your oranges and your lemons and your citrus fruits, hence the name citric acid. But this is why the Krebs cycle is actually called the citric acid cycle. We're going to be dealing with the citric acid, and we're going to be breaking it down and oxidizing it further. So then the Krebs cycle actually has 2 names that it can be referred to. So the second one is going to be the citric acid cycle, and this is why. So from here, we have the next step. What's basically happening is Krebs cycle is going to be an oxidation reaction. We're just going to be oxidizing a bunch of things. So from here, we're going to oxidize citric acid, and in doing so, we're going to lose electrons. So we're going to lose electrons, and to oxidize our citric acid, NAD+, again, NAD+ is going to be reduced. And this is the same idea of what I mentioned earlier. The electrons are released from citric acid, helping us reduce NAD+, because to reduce is to gain electrons. We're gaining these electrons from the reduced citric acid into NADH. The NADH is actually going to be used later to create ATP in the form of, or ATP in the electron transport chain. As well as doing this, we actually lose a carbon dioxide. So we're going from a 6-carbon compound to a now 5-carbon molecule. So 5-carbon. So 5-carbon molecule. You don't have to know the name. Again, this is going to be Bio 20. So we're going to be, what's resulting is a 5-carbon molecule from the oxidation of citric acid. And you're going to notice, to go from 6 to 5 carbons, we're going to lose a carbon, right? So again, this is the same idea that happened in the pyruvate oxidation. It's going to be released in the form of carbon dioxide. So a 1 carbon molecule. And this is where the carbon dioxide part of the cellular respiration formula actually comes in. We're losing carbon dioxide. So now our resulting 5 carbon molecule is actually going to get oxidized. Remember that the Krebs cycle is an oxidation process. We're losing electrons, so these electrons are going to be released. And that's going to actually help us reduce NAD plus into NADH once more. So again, we're creating NADH, which is again going to be really useful in our electron transport chain. And now we're actually going to want to create a 4 carbon molecule. So 4 carbon molecule. So we're going to create this 4 carbon molecule, and you're probably going to guess again. We're going to release more carbon dioxide. So we're going to release carbon dioxide. That 1 carbon from our 5 carbons is going to be lost through the form of carbon dioxide, dropping us down to a 4 carbon molecule, which you don't have to know the name of, but again, it's pretty cool. And here's the cool part. This is the only part of the cycle where ATP is produced directly. So 1 ATP, so we're going to, what's going to happen when we oxidize this 5 carbon to our 4 carbon? This is the only part of the Krebs cycle where we actually generate ATP. So you're going to notice we're going to create ATP here. We're going to break down ADP plus our phosphate to turn into ATP. And the whole entire cycle, each turn of the Krebs cycle, only is going to produce, one turn of the Krebs cycle is only going to produce 1 ATP per cycle. It's small, but it's still important. And when we're doing our ATP net calculations in the end, this is important because our Krebs cycle doesn't actually produce that much energy. Where most of our energy is actually coming from is the electron transport chain. So our last final step is going to be regenerating our starting compound. So remember that we started with this 4 carbon compound. This 4 carbon compound that we've created here isn't actually the same, right? So we're going to have to regenerate it. Now we're going to go back to this 4 carbon compound to restart the cycle. So in order to do this, what we need to do is we need to oxidize this, oxidize it again. And in order to do that, we need to reduce FADH, or FAD, a molecule called FAD, to FADH2. Remember, if we're reducing, we're losing electrons, so this 4 carbon molecule is going to release electrons. It's going to help us reduce FADH to FADH2, which is used in the electron transport chain for energy as well. In order to get us back to that original starting 4 carbon molecule, we actually need to reduce NADP plus, or NAD plus, once more into NADH. So these reactions help reform our original molecule, and just for this case, so it's not confusing, this molecule is going to be called OAA. That's the starting molecule, right? This 4 carbon is not OAA. To regenerate that, we actually need to go through one final process of reducing NAD plus and FAD to FADH2 and NADH, which are later used in our electron transport chain to give us a bunch of energy. So you may be thinking, like, why does this matter? Like, this is cellular respiration. Like, there's barely any energy being created, right? But here's the trick. The real power of the Krebs cycle lies in NADH and FADH2 that were made. These molecules carry high energy electrons to the next step. Because remember, we're oxidizing these big compounds, like from citric acid to this 5 carbon compound. We're oxidizing. And in doing so, we reduce NAD plus to NADH. Remember, reducing power. When we reduce, we're actually gaining energy. We're energizing these compounds. We're re-energizing this NAD plus to create NADH. High energy. Same goes for FADH. We're energizing it, right? We're energizing this FAD to create FADH2. And at this point, they're high energy, and these molecules carry high energy electrons to the next step, the electron transport chain. So in short, basically what the Krebs cycle is doing, it's effectively a high efficiency generator, fueling the electron transport chain with electron carriers, all starting from that small little molecule, carbon molecule, Acetyl CoA. We're being super efficient here. We're turning Acetyl CoA into a bunch of different ways that we can create energy in our electron transport chain. So if we were to look at the input versus the output, since this whole process has gone a little messy, as we probably noticed, as you'll see, we're inputting Acetyl CoA. So this is just a nice little summary. Our input is Acetyl CoA. What's outputted is going to be carbon dioxide. So how many carbon dioxides are released here? We have 1, 2. So 2 carbon dioxides are released. So we have 2 carbon dioxides. And remember that this process actually is going to happen twice. So the Krebs cycle is actually going to happen twice. Because remember, 1 pyruvate creates 1 acetyl CoA. In glycolysis, we created 2 pyruvates. So we're going to create 2 acetyl CoAs. Meaning our Krebs cycle is going to happen 2 times. So we're actually going to have 2 acetyl CoAs. And instead of 2 oxygens, we're going to have 4. Or 4 carbon dioxide produced. In terms of our input, what we need is, let's look at the NADHs created. So NADHs that we're inputting. We're inputting this NAD plus over here, and NAD plus. So we have 3 initial NAD plus that we're putting in. What's outputted is going to be 3 NADH. But again, remember that this happens 2 times. So we're actually going to have 6 NAD plus turning into 6 NADH. In terms of FAD, we're going to put in 1 FAD, and that's going to give us 1 FADH2. 1 FADH2. And again, that's only per 1 acetyl CoA. We actually have 2 acetyl CoAs. So we're going to have 2 FAD and 2 FADH2. And that is what the in and out of this process of the Krebs cycle actually looks like. Hey everyone, thank you guys so much for watching. This video, hopefully it helped in helping you understand the Krebs cycle and the process of glycolysis. Again, it's a brief video just to cover and be quick on what actually happens in glycolysis and the Krebs cycle and why we actually do it. So if you learned anything from this video, that is a positive for me. So if you guys like this, leave a like, and I'll see you guys later.